we resume our journey of the anther. In the last class, I talked to you about all the 4 different wall layers of the anther. We were talking about tapetum. Now coming to the functions of tapetum, tapetum is the most important wall layer, like I told you, because this performs the function of nutrition to the developing pollen grains. So it is primarily nutritive and secretory in function. Cells in tapetum, they have abundant cell organelles, they contain a prominent nucleus, and they also are characterized by a dense cytoplasm. Cells here are often polyploid because of either mitotic divisions, formation of restitution nucleus, polyteny, or endomitosis. Now, tapetum is most important because tapetum exhibits a transfer cell-like activity. It is primarily transferring the nourishment which is coming to it from the mother plant to the anther locule where the microspores are developing. Tapetum also synthesizes and secretes molecules which are associated with the development of the pollen wall. Now, in a mature anther, tapetum generates completely. There are 2 types of tapetum, and these 2 types are basically it depends on the extent of their intrusion in the locule. That is, they differ on the extent of their intrusion in the anther locule. The first category is the amoeboid or the invasive or periplasmodial or the syncitial tapetum. This is characterized by early breakdown of inner and radial walls of the tapetal cells. The second type of tapetum is the secretory or glandular or parietal or non-syncitial tapetum. In this case, the cells maintain their position and secretory nature unlike that of amoeboid tapetum. Now talking about the amoeboid tapetum. Amoeboid tapetum has derived its name because it somehow functions like an amoeba. You would recall that amoeba engulfs its food material, and here also tapetum, it breaks down early and the protoplasm extrudes into the sporogenous cavity to provide nourishment to the developing microspores or the microspore mother cells. Tapetal cells, they fuse amongst themselves to form a tapetal periplasmodium, and this periplasmodium, it moves into the anther locule and like amoeba, it engulfs the microspore mother cells. Now, how is this periplasmodium formed? Basically, the cell walls, they get dissolved, and they get dissolved by the action of hydrolytic enzymes which are secreted by the dictyosomes, which are very much present in the tapetal cells. So, with the dissolution of the cell walls, all the protoplasm, it combines and there is a periplasmodium which is formed, and this periplasmodium moves into the anther cavity engulfing the microspore mother cells. Now, amoeboid mass itself produces the precursors for pollen wall material. Now, as the periplasmodium moves into the anther cavity and it is directly engulfing the tapetal cytoplasm, providing nutrition, the volume of the locular fluid in those species which exhibit amoeboid tapetum is much reduced. There are abundant plasmodesmata connecting the tapetal cells which are seen in an amoeboid tapetum. The number of ribosomes and dictyosomes in the cell cytoplasm is relatively reduced. Now, in the next type of tapetum, we will study that there is formation of Ubish bodies, but in amoeboid tapetum, the Ubish bodies are not present. Amoeboid tapetum is seen in Alisma, Butamus, Tradescantia, and many other species. In this particular picture, you can see a transverse section of an anther with amoeboid tapetum, and this amoeboid tapetum, which is darkly staining here, you can see it is engulfing these spore tetrads. So, you can see the spore tetrads which are present in the microsporangial cavity, and these microspores are engulfed by the tapetal periplasmodium. So, what is happening here is the tapetum has lost its own position and it has moved into the anther locule, and this is a characteristic feature of amoeboid tapetum. The second type of tapetum which we will be talking about is the secretory tapetum. Now, in contrast to the amoeboid tapetum, the tapetal cells, they remain intact as they supply nutrients in the form of secretions to the developing pollen grains. So, they are not fusing here with each other, nor are they moving into the anther locule, but they remain in their own place, and from there only they secrete nutrition into the anther locule and eventually they disintegrate. There is no secretion of hydrolytic enzymes involved here, which was involved in amoeboid tapetum. Now, a characteristic feature of secretory tapetum is the presence of Ubish bodies. Now, these Ubish bodies are a characteristic feature of secretory tapetum. These Ubish bodies basically, they are present in cells which have secretory tapetum, and they originate as small lipid molecules, and these small rounded bodies are called as the pro-Ubish bodies. Once they get coated with sporopollenin, they are called as the Ubish bodies. Now, since these cells, they are giving in their secretions to the anther locule by being present at one position and not moving into the anther locule. So, those species which exhibit secretory tapetum, there the locular fluid volume is much larger. There is very high number of ribosomes and also endoplasmic reticulum in the tapetal cell cytoplasm. Such tapetum is observed in rice, in wheat, in Helleborus, Brassica, Arabidopsis and many others. In this particular picture, you can see that there are elongated cells with many nuclei which are present as the innermost layer of the anther wall, and in the sporogenous cavity you can see that there are developing microspores. The tapetal cells, they remain in their position till the maturity of the anther, and this is a secretory tapetum which you see here. Now, this is a transmission electron micrograph of small rounded bodies which are called as orbicules or also Ubish bodies in the microspores. These orbicules or the Ubish bodies, they line the inner tangential walls of the secretory tapetum. They are smaller than 1 micron in diameter, and they can frequently fuse into larger aggregates. Like I just mentioned, they originate as lipid droplets and they get coated with sporopollenin. And once they are coated with sporopollenin, they are extruded into the anther locule, and then they develop simultaneously with the growing pollen exine. They play a major role in pollen wall formation. Now, I want to mention here that these orbicules or the Ubish bodies, like it is mentioned, that they originate as lipid droplets. Now, they try to aggregate towards the membrane of the tapetal cells. They aggregate over there, and then they are also lined by the ribosomes, which appear as the spokes of a wheel. So, the ribosomes along with these lipid droplets, they actually have to move from the cytoplasm of the tapetal cell into the space which is present between the tapetal membrane and the tapetal wall. So, the membrane is convoluted and these pro-Ubish bodies, as they are till now called, they move into the space between the tapetal membrane and the tapetal cell wall. So, while they occupy this space, they get coated with sporopollenin, and from now on they are called as the Ubish bodies. So, a pro-Ubish body, when gets coated with sporopollenin, it is called as the Ubish bodies. Eventually, the tapetal wall disintegrates and these Ubish bodies, they move into the anther locule, and here they aid in the development of the pollen wall and they play a major role in the pollen wall formation. Now, just a recap of the two types of tapetum that we have just talked about, the amoeboid and the secretory tapetum. In amoeboid tapetum, the tapetum breaks down early. In secretory tapetum, the tapetal cells remain intact till the maturity of the anther. In amoeboid tapetum, the cells fuse to form a tapetal plasmodium, whereas no such plasmodium is formed in the secretory tapetum. Amoeboid tapetum cells extrude into the sporogenous cavity, like I had mentioned that they move into the sporogenous cavity and they engulf the developing pollen mother cells, while in secretory tapetum the cells do not extrude into the cavity, they remain in their original place. Amoeboid tapetum engulfs microspores like amoeba. In secretory tapetum, cells do not engulf the microspores. In amoeboid tapetum, hydrolytic enzymes are secreted, which eventually are instrumental in the dissolution of the cell walls. In secretory tapetum, no hydrolytic enzymes are secreted. While in secretory tapetum, Ubish bodies are present and secreted, no Ubish bodies are formed in amoeboid tapetum. Now, these Ubish bodies are involved in the exine formation of the pollen grains. In amoeboid tapetum, since there are no Ubish bodies, the amoeboid tapetum has no role in the exine formation. The number of ribosomes is very few in amoeboid tapetum, whereas the number of ribosomes is very large in case of secretory tapetum. Now, functions of a tapetum. Like you have understood that of course tapetum is involved in supplying nourishment to the developing pollen grain. It is the most important wall layer of the anther as it controls the reproductive development, because tapetum is the one which is an intermediate between the gametophytic and the sporophytic tissue of the anther. Tapetum also contributes to the pollen wall proteins which are involved in pollen pistil interaction, and sometimes these proteins they also call pollen allergies or hay fever. Tapetum induces callose plug formation in incompatible pollinations. Now, another thing which I want to mention here is that tapetum is the one which secretes a callose wall which separates the microspores in a tetrad. So, in a way, all the microspores they grow and develop independently while being enclosed in a callose wall. Now, this callose wall has also to be degraded when the microspores become mature. And how is it degraded? It is degraded by the action of a callase enzyme, which too is secreted by the tapetum. So, if the callase enzyme is secreted early, then the microspores have not fully developed, it can lead to the sterility of the microspores. Or on the other hand, if this callase is secreted late, then also the microspores have passed their time of fertility and viability. So, tapetum is responsible for the fertility of the microspores. Like we just discussed, that tapetum produces the oobish bodies, and these oobish bodies are coated with sporopollenin, and this sporopollenin is a precursor to the pollen exine. So, tapetum is involved in the growth and development of the pollen wall or the pollen exine. It provides pollen coat substances. There are pollen coat substances like the pollen kit and the tryphene. It gives rise to temporary wall of callose, which separates the microspores in a tetrad, which I just explained to you, and at the same time it secretes callase enzyme, essential for release of pollen grains from the tetrad. Normal development of tapetum is indispensable for completion of male meiosis. So, you would have understood that this tapetum is really important for the growth and development of the pollen grains, and pollen grains being the carriers of male gametes, tapetum plays a very important role in the reproduction of angiosperms. Now, the pollen grain or the male gametophyte. Now, the development of this male gametophyte is divided into two phases, which we will be taking up in the next class. But just to give you an idea, the phase 1 comprises of microsporogenesis. Now, microsporogenesis means there is meiosis which occurs in the microspore mother cells, and there is formation of microspores. Phase 2 is when microspores, they develop into pollen grains, and then the pollen development occurs with the formation of two male gametes. So, when there are two male gametes formed, this process is called as the microgametogenesis. So, the development of male gametophyte is divided into microsporogenesis, wherein meiosis takes place, and there is microgametogenesis, where mitotic divisions take place. Now, last but not the least here is the dehiscence of anther. Now, we have studied the different wall layers of the anther, and we have also talked about the microsporangia. There are 4 microsporangia which are present in a typical anther, and each microsporangium contains the microspores. When the microspores are mature, when they are fully developed, then we refer to them as the pollen grains. So, actually pollen grain in its stages of growth and development is referred to as the microspore. A spore mother cell undergoes meiosis, and then the microspores are formed. These microspores are initially present in a tetrad, that is, when meiosis happens, there are 4 microspores which are formed. Now, once the callase acts on the callose, which is binding the microspores together, these microspores are liberated into the anther locule, where the further growth and development of these microspores takes place. And this is the time where the locular fluid is important. If there is more locular fluid present, like in those species where there is a secretory tapetum, then this locular fluid also adds and helps in the growth and development of the microspores. Eventually, when the microspores have developed their walls and they are fully mature, they are called as the pollen grains, and this is the time when the anther has to dehisce to liberate these pollen grains. Now, what is dehiscence of anther? Dehiscence of anther is basically, it is the opening or the breaking open of the anther to liberate the pollen grains which are present, so that fertilization can take place. Now, in this particular picture, you can see on the top that there are 4 anthers which are present. In one, there is longitudinal dehiscence, that is type 1 of anther dehiscence. Here, the suture, the opening of the anther is along the longitudinal axis of the anther. So, the anther splits open along the longitudinal lines and the anthers are liberated. The second one is the transverse type of dehiscence. You can see that in the middle of the anther, there is a suture which will split open and liberate the pollen grains. Third one is poricidal, which you see that there is a pore which is present at the top of the anther here, and it is through this pore that the pollen grains are liberated. This is commonly seen in grasses, and you can see in figure B over here. Then there is valvular dehiscence, that is when the pore, it is covered by a flap like covering, that is what we call as the valve, and this is valvular dehiscence. Now, the extreme right figure which you see here is of longitudinal dehiscence, which is seen in lily. So, there are primarily 4 types of anther dehiscence: longitudinal, transverse, poricidal and valvular. In longitudinal, like I just mentioned, it is seen in brassica and lily. Dehiscence suture is parallel to the longitudinal axis of the theca. In transverse, dehiscence takes place at right angles to the theca, and this is commonly seen in species of Ocimum. Poricidal, as the name suggests, is by means of a pore at one end of the theca. Now, in this particular one, you can see in grasses, it is figure B which you see over here, and this pore could be on either end of the theca depending on the species. The last or the fourth one is the valvular, where dehiscence occurs by a pore, and this pore, unlike grasses, is covered by a flap of tissue and is referred to as a valve, and this is seen in Lauraceae. Right. So, today we have talked about the anther in detail. We have talked about the different wall layers of anther, wherein you have the epidermis, you have endothecium, you have 2 to 4 middle layers, and of course there is tapetum. Also, we talked about the different types of tapetum. We have talked about the different functions of each of the wall layers of the anther, and of course the sporogenous tissue, which we will be taking up in the next class. I told you about the different types of anther dehiscence, like I told you in this slide, that now we will take up the development of male gametophyte in the next class, which will be divided into microsporogenesis and into microgametogenesis. And with this, we finish this class here, and thank you very much for your patient hearing. Thank you.